Good afternoon, everyone. I am Morgane Zimmer, a PhD student from the INL laboratory in Lyon, in France. So I want to present to you not an application for lab on chip, but explore the idea of using another materials for the fabrication of this kind of device. So lab on chip are microfluidic systems that enable the manipulation of fluids or micro objects. By miniaturizing classical laboratory function, we can bring down the cost of application, such as for environmental or food analysis, for drug development, or even in the bioanalysis, as we are studying today. So one other advantages of lab on chip is that it's portable, so we can bring it outside and on the field in low resource settings. And the final advantage for application in the healthcare domain is that due to the size of the microchannel, it is possible to do experiment on single cell scale, and this is very useful. However, all the materials that we use to produce this lab on chip are from the petrochemical industry, as PDMS and thermoplastic. And moreover, most of the applications are only single use, so we have to throw away the device after each application. And this contributes to the growing amount of plastic waste in produced by our society. So in my project, we want to propose new materials, which is Chitosan, a biopolymer, to fabricate this lab on chip. So Chitosan is industrially produced from chitin, the second most abundant polysaccharide on Earth, just after cellulose. It is also, we can obtain it by valorizing waste from the food marine industry. And if we consider only shrimp, we can obtain between 6 and 8 million tons of Chitosan per year. So we have a sufficient amount on Earth. And more interestingly, it is biodegradable. So we will reduce the impact on the environment. Now, if we look to criteria to create this device, we first need to be able to fabricate a film that is thick enough to have a microfluidic system, so more than 200 micrometers. We need to be able to microstructure the polymer film to create the channel inside it. We need it to be watertight, so there is no leakage of the fluid through the materials. Then for our application, we want to be able to manipulate cells, so it needs to be biocompatible. And it needs to be transparent, so we can observe the cells during their passage through the device. Now, for the sustainability part, we need the materials to be biosourced and biodegradable. So if we look to Chitosan from previous research, we know that it is biosourced and biodegradable. It has also film forming properties, but it has been mostly used to create very thin layers of Chitosan. And it has also been used to as a cell culture media, so it is biocompatible. Now we just need, from this potential of Chitosan, we just need to find protocol to fabricate the lab on chip. And this is what I did in my project. So from Chitosan powder, I will present to you the protocol to fabricate a microfluidic system. We first need to dissolve the powder in solution. However, as the Chitosan it is insoluble in water. So we need to dissolve it in acidic solution. By bringing down the pH, we will protonate the amino group of the Chitosan, which make it soluble. Then we centrifuge the solution to remove any bubbles, and we dry it in an oven at 50 degrees. After this, I characterize my films. So you can see that the thickness of the film would depend on the initial Chitosan concentration, as well as the Chitosan that I use. Depending on the molecular weight, I will obtain different films. You can see here a picture of these different films. And by compromising the properties, so the thickness and the transparency of the film, we conclude that it was better to use a medium molecular weight due to the color of the film and the thickness that we obtained. And this was the property that we studied, the film that we studied after. we have a transparency of an average transparency on the visible range of 75% and a Young modulus of 6.4 gigapascal. Now the film, if you remember, is still soluble in water. So we need to add another step in order to be able to use it in a microfluidic system. So we use a neutralization step. We immerse the Chitosan film in alkaline water. We try different concentration of sodium hydroxide and different immersion time in this alkaline solution to check the properties that we obtain. This will deprotonate the amino group and make it insoluble again. Then the film is rinsed to remove any molecule left. And finally, it is dry once again to obtain a film. Now, if we want to characterize the property of the different neutralization method, we measure the swelling of the film, how much water it absorbed, by comparing the weight of the film when it's dry with after spending some time in deionized water. So here you can see, depending on the time spent in water, the swelling of the film. So firstly, you see the highest swelling rate is for films that are not neutralized. And after 30 minutes in water, they even start to dissolve. So we have no point further. And you can see more clearly, if I zoom in here, you can see that depending on the concentration of sodium hydroxide and the time spent, if we increase both the concentration and the time spent in the solution, we will decrease the swelling, which we want. We want to have as low swelling as possible. So this is why we started using the concentration of 1 molar of sodium hydroxide, where we left for 45 minutes the film in this solution. Now we obtain a swelling of 49% with the same transparency as before the neutralization, with a slightly higher Young modulus and a thickness of 330 micrometers. We also have a hydrophobic contact angle of 99.5 degrees. Now we have a film that is not soluble in water anymore, so we need to microstructure it. We choose to use the microdrilling method, where you just need to drill the shape that you want on top of the Chitosan film. You can even drill holes through the Chitosan film. One advantage of this method is that you can vary both the size, the width, and the height of the channels as you want on the system. You can obtain several of them. Here, for example, you have 3 channels with different sizes, from 250 micrometers to 1,000 micrometers wide. We characterize the dimension by optical microscopy and profilometers to verify that we obtain the dimension that we wanted. And now we still need to seal the device. So we choose to use a dry resin that we laminate on top of the Chitosan film. Then we press it on another substrate to bond it. And we insulate the resin with a UV lamp to cross-link the resin and fix the system. So we managed to bond the Chitosan film to either a glass slide or another Chitosan film. And we verified the water tightness of the system by injecting aqueous solution that are colored with a food dye on both of the system. Here is another more complex example of the kind of system that we can obtain from Chitosan. It is a Chitosan film that is bonded to a glass slide, and we injected in 2 different inlets, both red colored water and blue colored, with a flow rate of 1 milliliter per hour. You can also observe that Chitosan film is highly transparent, so it's enough for our applications. Now, if we look back to the criteria that we specified at the beginning, so we first managed to make the first microfluidic system in Chitosan, and we managed to have a film that is thick enough for microfluidic application, and it has a 55% transparency. We have a watertight systems, even if there is a slight swelling left over. So we are working on this to improve the property of our film. We managed to microstructure the device with microdrilling, which is a highly flexible method, but it is limited in resolution due to the size of the microdrill that we use. And all in all, this is the first step for a more eco-responsible lab-on-chip fabrication. and we are hoping to include more complex functions in the future. So I want to thank you all for your attention, as well as all my team back in my laboratory, and my funding from the LabX IMUS.